All right, good evening. Let's do some questions and answers for A World from Dust, chapter 7 through 9. So, this is the third quarter of the book, and we're getting pretty close to humans. We're getting to brains, but we haven't quite gotten to humans yet. And so, we have lots of questions that are getting on to the point of closer to where we live. We're moving away from the Earth and sort of toward us. But I do want to start with the Earth because one of the things that was there is what are those weird pink ponds that you see in San Francisco Bay? Here's a picture of them, and there's actually, I've always called them salt ponds. I hope I didn't actually call them salt ponds there, but maybe I did. If I did, this person would be upset with me because they actually are called crystallizer beds. Okay, whatever you want to say. If you want to listen to this, you can find out a little more about exactly what they are. But this is what they look like for those of you who have never flown into San Francisco, flown over the bay, and seen the ponds there. Maybe you've seen them somewhere else. I'd like to know. So this is where we have if you lived in San Jose, then you would see them all the time. We have a comment about that. The question is related to sulcatone that the mosquitoes are attracted to. And is it an interspecies volatile, and where does it come from? Yes, it's very much interspecies. In fact, it's interkingdom. It's actually found in species of animals as well as species of flowers. And in fact, there are different mosquito species that are attracted to it that have nothing to do with being attracted to humans at all. They're just attracted to the flower. This is probably one of the reasons why the mosquitoes are so into it, right? It's probably just a very good attractant. I'll show you the structure in just a second. Someone with sweet blood will attract all the mosquitoes, so that's kind of thing. And being in close proximity to a capybara also seemed to help. I think there could be a rigorous controlled study. I'm not sure this would get an NIH grant, but maybe an NSF grant. I don't know. But sweet blood could be related to sulcatone levels. It's very possible because it is a major signaling mechanism. It has a receptor, and now we know more about it. We actually know the receptor that it goes to. And there's both the animal mosquito and the plant mosquito are both attracted to sulcatone. So this is something that's been passed down. And a lot of flowers will have this. You see that it's a very small volatile molecule. I have the name right here. 6-methyl-5-heptane-2-one is what it is. And it's very clearly a volatile thing. You can study it with GCMS. There's all sorts of interesting studies about it now. So it's probably not the only thing, but it's a major thing. And it's, they talk right here about how there's this plant sensing mosquito that goes after sulcatone as well. So this is an article you could find out more about. The interesting thing that it claims is that DEET will inhibit the receptor's binding site. And I don't know the structure of DEET, but I'll bet it has some kind of chain structure. It's volatile like this. Why not capybaras? I mean, they're like guinea pigs. They're closely related to humans in some particular ways, and I could very well see that they would have maybe they release sulcatone for some reason. Maybe they release it for no reason at all, just a happenstance. Again, it's plausible. I'll take it. So the next question, we're moving up from small molecules to heat shock proteins now. We're moving up the scale of things. And what about the structure allows them to continue to function in stressful conditions? Why don't they unfold like other proteins? Well, it's because it's their job. They can actually be stabilized, and it's actually pretty easy to make a protein more heat stable. In fact, when they first developed protein design algorithms, the first thing they did with them is they tried to design proteins that were more heat stable. And they actually ended up being so successful at that the proteins were too stable. They had to actually destabilize the proteins because it was so easy to make a protein that's more heat stable. This also attracted a lot of interest from pharmaceutical companies. If you could make a protein that was shelf stable with a couple of mutations, it would work better. Unfortunately, the same mutations seem to affect how the proteins work in general, but if you have evolution working on both function and structure at the same time, evolution has no problem evolving heat shock proteins that will be more stable and will also function to either bind hydrophobic portions as a small protein or be a cage around the hydrophobic protein as a big protein. And so that's the main thing. There are two kinds of these proteins. There's really small ones that just go after segments of a protein. They're like little tugboats. Then there's really big barges that will actually take the entire other protein into it. And it turns out there's even some that are small, and then under certain conditions they form big cages. So 24 of them get together and form a cage. I found a picture of one of those, and I just think these pictures are really cool. This is a small heat-shocked protein, but it comes together. 24 of them come together to make this big cage-like protein. And I believe that I know that this can protect crystalline, which is a relatively small protein. So I think it works as a cage that the protein goes inside it. They found a mutation that actually makes it work better, but you can see right here that's what the R107G mutation is. At room temperature, you have a pretty big difference between the two. When you heat it up to 60 degrees, actually the R107G works better for its chaperone function. And what you see right here, it's a better cage, and we don't know exactly why it's better yet, but there's some ideas because we can see that it definitely has a different structure. And yet, these are definitely still, both of these, both wild type and R107G, are definitely still holding together when they're heated up to 60 degrees Celsius. So, yeah, it's actually not that hard to make a heat shock protein. In fact, there's an argument that the cell wants to make proteins that are just mildly stable in general, and then it can make these special heat shock proteins that are extra stable. And the cell doesn't want proteins to be rocks, so it's a lot of normal proteins are just barely stable enough because the function is that they need to be stable enough to do their job, and then they need to get out of the way or be cleared. So the idea of superstability is not something that life is usually going for, unless it has a reason. And with heat shock proteins, it has a reason. So hopefully that clears some of that up a little bit. Now we'll continue to move through, and there's things about chlorophyll. And yes, it's really important to keep in mind chlorophyll is a metalloprotein. That technically chlorophyll is the metalloporphyrin, but chlorophyll-containing proteins are metalloproteins. It's easy to forget because the magnesium is a weird metal, relative to the other ones. It's not a transition metal, but it's definitely still a metal, and it's very important to the whole course of natural history. Could this magnesium be the same? And it actually could be. The thing about chlorophyll is it's relatively fragile. Like compared to other molecules, it's relatively fragile. But that's actually good because you can break it apart and maybe get the magnesium if you need it. Or maybe the chlorophyll, the empty chlorophyll, can do something for you. So the magnesium itself wouldn't look green because outside of the chlorophyll, the magnesium doesn't look green. But the chlorophyll itself would probably turn colors as well. It might be more yellowish. And I'm thinking of my friend who would make carrot juice a lot, but she would actually start to turn a little bit orange because in that case, I believe it's mostly the beta-carotene, which is a different pigment, but it would act much the same. The beta-carotene is fat-soluble, so it goes to the fatty layers under the skin, and it gives the whole skin an orange hue, and you literally will look orange. So I know it works for beta-carotene, and it seems to make sense. Chlorophyll is a little less fat-soluble, but it still seems like the same kind of thing might happen. It's just it probably wouldn't be green because when the magnesium falls out, the chlorophyll turns more yellow. So it probably happens less. But I don't see why it couldn't happen. I think it's a reasonable question to ask about how could this happen. You can actually, all these questions, I'm just guessing. You can always follow this up and look for things yourself. So now, I was searching for this and looking for it, and I found out that apparently there's a TikTok craze where chlorophyll will clear up your skin. Like you drink chlorophyll and your acne goes away. And there's lots of people saying that it's there, and I don't know. I haven't tried this. I don't really want to drink chlorophyll, but I guess there's grosser things to drink. As far as trends go, it's probably a pretty innocuous one. So it's worth trying, I don't know what the mechanism would be, but if it goes to that fatty subcutaneous layer, who knows? Maybe it could try something. Maybe just the placebo effect would work. I don't know. If it works, in my opinion. And I found out all these things. Now, this is from like a health magazine source, so I'm not sure how much these are. but I think it's really interesting that they had a list of chlorophyll's uses. It's actually used to treat pancreatic disease. I don't really know how or why. And it's actually used to inject it to the body to help the wounds caused by cancer to heal. And is that the magnesium, really? I can't imagine the chlorophyll itself does that unless it fits into the heme binding site. Anyways, I have more questions than answers here, but it does look like chlorophyll is a very useful protein. And in fact, the old experiment that we got our protein unfolding experiment from is a chlorophyll experiment. It was a chlorophyll protein, and that's why it was green, and it would change absorbance when you unfolded it. But the thing about the skin and chlorophyll, again, it looks like there's something there. There's a study on women that the chlorophyll extract improved wrinkles and elasticity. I believe this is a topical application. And then there's topical chlorophyllin, which is a water-soluble version with added sodium and copper for easy absorption. I don't know about that, but it significantly improved acne. And basically you can get chlorophyll from spirulina powder, which is exactly what we went to the health food store and bought when we wanted a green protein for the unfolding lab. So chlorophyll A is green with a blue tint, and chlorophyll B is green with some yellow. I can actually show you the separation chromatography of these. It's a very common chromatography experiment to separate out the green pigments and the orange pigments in plants. It's you can do it with like a potato column. It's very easy to do, to separate out these pigments, and it looks beautiful. So there's chlorophyll is only, and I want to remind you, chlorophyll is 2.6% magnesium. So yes, it's the same magnesium, but the magnesium is technically just 2.6% of the weight of a chlorophyll extract. Even less if you're talking spirulina, because spirulina is a whole protein, or maybe even a whole organism that's carrying the chlorophyll for you. Okay, so one last observation and question is that there's a lot of issues early childhood with the brain and its development, and your mom's focus is learning to retrain the brain with developmental problems and effective forms, and talking about the various medical issues with that. So learning the brain is particularly susceptible to ROS damage. Would these issues be related to ROS? And the answer is almost certainly, in a sense. And the question is, in what sense are they related? How closely are they related? And is it the kind of relationship that can actually help you do something? You know, if a lot of the brain damage that can happen from the ROS is probably irreversible. Once it happens, and it's kind of like entropy, it's hard to put that back into the bottle. It's hard because the neurons stick around and don't regenerate. Once they're damaged by the ROS, then they continue to be damaged. So there are ideas, and I'm sure people have even tested some of these, and but it's a very complex system, and ROS are just one part of it. Could complex similar to the leaf complex, and then we have manganese, so magnesium with the leaf complex, or more manganese. Manganese phosphate, I believe, was the protective chemical. Now, I think manganese phosphate is probably toxic in the concentrations that you would need, and you probably don't want that in your brain anyways. I don't know if manganese might be a good shield for more or less central tissues, like, I don't know, if it's a more extreme and more regenerative, because the manganese would work against the ROS, but then it would probably be doing other stuff as well. But again, it's not a bad idea. It's just that these things are so complex that we have to test the idea. Maybe it would be a good idea for a grant someday or something like that. But the other thing it reminded me of is Nick Lane, who's written a lot about origins of life. I actually quoted him in the Origin of Life chapter because he and his colleague had a really good theory about how pH gradients could form in hydrothermal smokers. He also talks about ROS and actually has a whole book about the citric acid cycle that I recommend called Transformer. And it's really good, especially for someone who's been through biochemistry, because you can sort of read through quickly all the stuff that he talks about with biochemistry, and you realize how much you learned in biochemistry. But he talks about ROS, and he does have particular theories about how ROS works. he thinks that antioxidants, like taking an antioxidant, won't really do anything for the ROS. And I kind of agree with him. I'm usually of the opinion of it can't hurt as long as you're doing it in moderation. But I also, just think of it as a secondary reason to like drink green tea or because of its antioxidant effect. There's probably other effects that are more important. Because if you're ingesting something, it's got many walls to get through before it gets to, especially the brain. But the interesting thing about him is he talks about mitochondrial damage as being not just for the brain deteriorating over time, but for the entire body deteriorating over time. The idea is that the mitochondria leaking ROSs are causing some kind of constant damage, and even our response to the damage might be suboptimal, but ultimately it is mitochondria and ROSs that are causing aging in this idea. So you see how this is a systemic, not just the brain, but the whole body can be affected by ROS. But it also shows that probably if it's such a complex and widespread issue, with such a fundamental thing as the mitochondria, we really depend on them, so we don't want to mess with them that much. You can see how probably it doesn't have a silver bullet like just take a bunch of manganese phosphate and then everyone will live to be 200 years old, things like that. Maybe that was Methuselah's secret. But in a small scale, it's a thing to try, but I just want to point out the ROS, because it's so widespread, because oxygen is so essential, it's not just a brain thing, but it has to do with the whole body. And so these are complex things, but I would love to see, some kind of thing where you work your way up to prove that it's safe, and then you try to give it to maybe animals, and then you try it on humans if it proves to be safe for everyone. And again, if you're going to take a supplement, choosing one with manganese, I don't even know if they are in the supplements, but I don't know if we humans use manganese enough for it to be a supplement type thing. But if there is one, again, there's possibility of toxicity. It can hurt, really. But I'm of the opinion of try stuff like the chlorophyll. And if you try stuff with a scientific basis, that's a good reason for trying it. You just got to remember that the scientific basis, even if it works, the scientific basis still might be misunderstood or wrong. But it's a good reason. I think it narrows your chances, it stacks the deck, and it makes things more worth trying if you have just a slight likelihood boost from you actually having a scientific theory that might work. I've tried lots of things. There was one thing that I tried that I got from a study about mice, sandalore. It's like a essential oil or something like that. And I eventually decided it wasn't working, but it wasn't that bad. I didn't have any bad effects. So just try stuff, but turn around if it's not working, but maybe it'll work. And maybe it will be just a coincidence that it worked and you just keep doing it. But if it works, it works. So yeah, live experimentally, but always keep an eye out, because even the best scientific theory is only a piece of the puzzle, and we are very complex organisms. There can be side effects and surprises that happen a lot. So great questions. Thank you for your comments and questions. We have moved up through life, and now we will have 1/4 of the book left. That quarter of the book is shorter, but it's also, in my view, it's more fun, because we actually are going to get into culture in chapters 11, the history of culture, and our current culture, our philosophical questions about how do we do with all this in chapter 12. So I look forward to one last Q&A when you finish all that. All right, thanks for this, and I will see you in class.